But before we discuss about the Mendel's experimentation, we have to understand there are two type of traits. One is called as inherited traits and one we call as acquired traits. So we have to understand the difference between the inherited traits and acquired traits. The characteristics or the traits which get passed on from one generation to another generation, that is what we call as inherited traits. The characteristics that are inherited by the offsprings from their parents, that is called as inherited characteristic, like height, height of the progeny, color of the skin, or weight. There is also shape of the nose, etc., whether free earlobe or attached earlobe. These are the traits, some of the traits, those traits which we inherit from our family, which are passed from parents to their offspring or to the progeny. Such kind of traits are called as inherited traits, which are passed from one generation to another generation. These traits are called as inherited traits, whereas acquired traits are those traits which have been acquired by the organism over the period of the time, but it cannot be passed from one generation to another generation. For example, if someone somehow, like sun tanning, tanning of the skin color, sun tanning, it cannot be passed from one generation to another generation because it is a acquired skill. For example, if someone's parents are engineers or doctor, it's their acquired trait. They cannot pass it to their offspring. It's not necessary that whose father is engineer will become an engineer, or whose father or parents are doctor will become a doctor. They can be IAS, they can be IPS, they can be anything. Like the parents whose like the child whose parents are doctor, he can be engineer, he can be IAS, they can be IPS, anyone, anything. So it is not necessary that an engineer's progeny will be an engineer only, or a doctor's progeny or a teacher's progeny will be teacher only. So these are the acquired traits which has been acquired by the person over the period of the time, and these traits like knowledge, it cannot be passed from one person, it cannot be passed from one generation to another generation, right? So I hope that now it's quite clear to you that inherited traits are the one which can be passed on from one generation to another generation, and acquired traits are the one which cannot be passed from one generation to another generation, which cannot be passed from progeny, which cannot be passed on to progeny from their parents. So now the question arises is, before I discuss about the Mendel experimentation, I will that how the characteristics appear in an organism, how the characteristics, how gene control. Yesterday I have told you that the gene is the you can say basic unit of hereditary, but how gene control it, how, where, and how the traits are expressed. Now let me tell you, yesterday only we have discussed that within the nucleus there lies the chromatin material, and within the chromatin material there are the small segments which we call as gene, right? And gene is comprised of DNA molecule. is a double helix structure, which we have discussed yesterday, that gene is a double helix structure, right? Now, how the gene control the the particular like particular trait, how gene controls a particular trait. Let me tell you, gene has the ability, because it is made up of DNA molecule, it has the ability to produce the proteins. So gene produces the particular protein which will uh, gene has the property that it can control the synthesis of a particular protein which is required for a particular trait, right? So characteristic in the living organism depends on the protein that a cell can manufacture, right? In a cell, there are the gene in the nucleus, in the chromosome, there are the gene. Genes are the segment which are made up of DNA molecule. Now, gene has the capability to control the synthesis of protein. What has gene? What has DNA capability to control synthesis of a particular protein which is required for a particular trait. For example, the production of the protein for the height will be controlled by gene if that particular protein which controls the height is secreted by in large amount, is produced in large amount, then the progeny will be tall. If it is produced in a small amount, then the progeny will be dwarf. But who will control it? It is controlled by gene. The synthesis of a protein is controlled by gene. And where from the gene? The message for the synthesis of the particular protein is encoded in DNA. Right? Different proteins are needed to carry out the different functions, and then the production of this protein will decide the appearance of a trait in a progeny. So So it is the gene who controls the synthesis of a particular protein which is required for a particular trait, and then depending on this protein, production of the protein, whether the protein has been produced in proper amount, whether the protein has been synthesized in small amount, or whether the protein has been synthesized in more amount, will depend upon that what information is present in gene, that is decoded, encoded in DNA, right? So that production of that protein will control the appearance of a trait, that what type of organism, what type of that matlab, what how do the organism will look like, right? So now our next topic is the rules for the inheritance of the traits. I have told you that Mendel was the person who introduced the concept of that the characteristic, that the traits are controlled by the factor. Later on, these factors were known to be as the gene. So Mendel was the first person to conduct the experiment, that is on hybridization on the garden pea. And yesterday we have discussed about that. Why did he selected the pea plant? Why did he selected the pea plant? So we had to discuss about that. Let me tell you, for studying the hybridization technique, Mendel used the pea plant, and for that he first of all, to study that how the factors control the traits, he first of all cross-pollinated the plants, and then in the second step, he allowed them to self-pollinate. Now, what is cross-pollination? The pollen from the anther of a flower, right, were transferred to the stigma of the fully mature flower. Now, we have to first of all understand that there is a process that's called as emasculation. Now, what is the process of emasculation? Let me tell you one thing. There are the flowers which are bisexual in nature. The flowers are bisexual. That means that the flower has both one male part as well as the female part. The female and the male, right? This is a bisexual flower, right? Having both male, having both female and male part. Now, what is emasculation? To eliminate the process of self-pollination, because what is there that the anther from the same flower can transmit to the stigma of the same flower because both are present in the same flower. So there was a possibility for self-pollination, and to eliminate the self-pollination possibility, what a flower has to undergo before we cross-pollinate it, it has to undergo the process of emasculation. Now, what is emasculation? That is removal of anther, male part of the flower, without damaging the flower. Removal of anther from the bisexual flower without damaging the flower, without damaging the female part of the flower. That is what we call as emasculation. It needed to be done to eliminate the process of self-pollination. Because Mendel, first of all, in order to study about the law of inheritance, or what we call as law of inheritance, he first of all selected the pea plant and then allowed them to cross-pollinate. But for cross-pollination, he has to eliminate the process that there should be the self-pollination. So for that, he emasculated the bisexual flowers. That is, he removed the male part of the flower. Then he allowed them to cross-pollinate. That is, he brought the pollen from another flower and dusted it over on that female part of the flower which was emasculated. That is where the male part was already, we have get rid of the male part, and what was present in the flower was only the female part, right? This is called as emasculated flower. So that's what is known as cross-pollination. When the anther of some other flower crossed over to some other one, that is when there are two different flowers. When anther of flower A goes to the stigma, that is the female part of flower B, this is what we call as cross-pollination. And if the pollen grain from the same flower is transmitted to the stigma of the same flower, then this will be called as self-pollination. But we do not want, Mendel want to eliminate the process of self-pollination, so he emasculated the male part, that is anther, he emasculated it. Then what he dusted over he dusted the pollen grains of flower A pollen grain or the male sex cell of flower A on the stigma of flower B. That is the female part of the, right? So that there can be a pure cross pollination without any possibility of self-pollination. Then he covered it with the muslin cloth so that even if the pollen is traveling in the air, it will not hamper experiment. Right. So the crosses were respectively called, where he studied only one characteristic. That cross was called as monohybrid cross. Where he studied two characteristics, it was called dihybrid. Where he studied three characteristics, it was called as trihybrid. And where multiple traits were studied, that was called as polyhybrid cross. So for reaching on the law of inheritance, he studied the monohybrid cross. Now, what was the monohybrid? Yesterday I told you that the genes are written by using the letters, alphabets. If we have to express the dominant gene, then we will be using the capital, and if we have to express the recessive gene, then we have to use the smaller one, small letter. Okay. Now, let me tell you, to reach on the conclusion on the first law, he studied monohybrid cross. What was the monohybrid cross? Let me tell you, Mendel cross-pollinated homozygous tall. Now, there is one term that's called as homozygous, where both the allele or both the gene will be same. That will be called as homozygous or pure tall. Pure, right? Where I have told you that gene always work in pair, where both the genes will be same. They will be termed as homozygous pair. That will be told as GOUS. That will be termed as homozygous pair, right? And where there is a possibility that both the gene may not be same. They may be different. So this is not now homozygous. Now it will be called as heterozygous. So you have to learn all these terms. Now, what is homozygous? What is heterozygous? Now, to study, to reach on the first law, that is law of inheritance, he studied one. So the phenotype was height of the pea plant. The phenotype was height of the pea plant, that is the feature which he was studying was height. And let me tell you, to understand that how the factors or how the gene controlled it, he has performed the following experiment. He selected a pure tall plant and a pure short, or homozygous tall and then homozygous dwarf, right? So the genotype will be for the kid. For height, there will be either the plant will be tall or it will be short. So for that, what he took was the pure or homozygous tall and homozygous dwarf or short. Homozygous means where both the gene are same. Now, as you know that the sex cells are not to keep the number of chromosome constant that we have already discussed in the chapter reproduction. To keep the number of chromosome constant, the gametes have only are haploid cells. Gametes are haploid cells, whereas rest of the cells are diploid in nature. That means the gamete will have only one chromosome of each factor. That's one gene of. It means that gamete will carry either this T or the gamete will have this one, right? Because the gametes are haploid in nature. So now in order to obtain the first generation, that's first phylyl generation, that's F1 generation, he allowed it to crossbreed, right? Where he allowed that homozygous tall needed to be crossed with the homozygous dwarf. So what was the possibility? The possibility is that this T can cross with this. That is, there are four possibilities to have the gametes. That is, the gamete can have either this or this gene for information. And here also, let me tell you, these are the gametes which contain only which are only haploid in nature. The rest of the cells are diploid in nature. For each and every characteristic, there are two genes, but in the gamete, for each characteristic, there is only one. Now this gamete now there is a probability that this gamete will cross over with this one, or this gamete can cross over with this one. While crossing, they can use this. Okay, so there is possibility that we can get TT, TNT, TT and TNT, right? Let me show it to you that there is this possibility. And this generation, this is called as F1 generation, first phylyl generation. First phylyl generation show the characteristic of both the parents. As I've told you that the height of the pea plant is controlled by pair of gene. A plant is tall because it possesses factors for tallness, and plant is dwarf because it possesses those genes which are there for the dwarf. As I told you that the factors always occur in pairs, right? But in the gamete, there is only one factor out for each characteristic. And to obtain the F1 generation, the gamete has the chance that it can cross over with this gamete, and once the pollination will take place, so in the F1 generation, all will be tall. Now, how can we interpret it? That that here dominating gene is present in each So what will be the phenotype? The phenotype of first phylyl generation or F1 generation will be that all will be tall, but they are not homozygous tall. What they are heterozygous. Although all of them are tall, why? Because this is dominating gene which will decide the characteristic in presence of a non-identical gene. Dwarfness is not the dominating gene. It is I have told you that dominating gene is always written with the capital letter, and here you can see that the tallness is the factor which is the dominating gene, which will control that all which will show the characteristic, which will control the characteristic of an organism, and all the organisms which are produced through this process in the F1 generation will be tall because although they are not they are heterozygous, but still all of them will be tall. Now to study about the F2 generation. F2 generation, what he allowed to do then is that self-pollination, right? When the first generation plants were self-pollinated, what he obtained was the ratio. As I've told you that gametes will again be in the, there will be four gametes, once they